And we are here. I'm Dr. Williams, Chair of Medicine, and it is my distinct pleasure to introduce our research grand round speaker today. We will be hearing from Dr. Leslie Wolf, who's an clinical assistant professor and lab director in the Division of Infectious Disease in our Department of Medicine. She'll be speaking on roles of the clinical laboratory and biorepository in clinical research. A little bit about Dr. Wolf. Dr. Wolf Parrish actually was a Kentucky Wildcat back in the day, then where she got her undergraduate degree. She did her Ph.D. at University of Colorado, and then started her infectious disease postdoc research at North Carolina State. She then was on the adjunct faculty there at North Carolina Central University, and moved to a clinical assistant professor at University of Louisville in 2016. So she's been here not quite a decade and has worked her way up from assistant professor to clinical associate professor, where she is now. And we're so pleased to have you, Dr. Parrish. Thank you for coming and edifying us on lab for infectious disease.
Thank you all so much, excuse me, for being here. I am going to do my best to speak on behalf of the Division of Infectious Diseases Clinical Laboratory, but also cover for Dr. Subhadra Marimuthu, who couldn't unfortunately be here due to unforeseen circumstances. So I'm going to do my best to cover her part because she's director of our biorepository. Hopefully when you leave here today, there'll be some take-home messages that I leave with you. One is that we're a little different. I always feel like every lab I'm in is a little different because I've been in labs my whole career. But we're not a basic research laboratory. We're a diagnostic laboratory, which I'll get into. But we also support our division's clinical research interests, as well as other collaborators you'll see. We're just about willing to do anything that within our capabilities to support anyone at the university or outside the university. All right, so our diagnostic laboratory. This is, I think, feels what makes us really unique to the university is that we are CLIA certified. How many of you have heard of a CLIA certified laboratory? So what that means is because we are testing human and patients for diagnostic purposes, i.e. our results go back to the patient chart and someone uses that to make decisions. We are audited by the federal government or the representatives every 2 years to make sure our quality and a whole bunch of other things that fall under that umbrella are up to par so that we're providing quality assured laboratory tests. So our clients are healthcare providers from the UofL healthcare system, from the Norton healthcare system, CPA, which is sort of the what do you call it, the outpatient arm of Norton Healthcare, and even the VA hospital system to some extent. And we perform applied research. The laboratory was actually founded in 1971 by Dr. Martin Raff. It was very small at that time. I think it was Dr. Martin Raff, maybe a postdoc, and Dr. Ramirez, our former chief. So it started out very small. But in 1990, when the Clinical Laboratory Improvement Acts, the federal regulations came into being, Dr. Jim Summersgill was the director at that time. And so he was the one who made sure the laboratory was CLIA certified to do diagnostic testing. So when Dr. Summersgill retired, I became the third director of the Infectious Diseases Laboratory. And fortunately, I had about a year or so overlap with Dr. Summersgill. So it was wonderful to have that mentorship and helping me get adjusted to the division. So our ID laboratory team has, when I started, we had 5 people, including me. During the COVID pandemic, we expanded to 9 to meet the needs of our testing community. And overall, the ID laboratory supports service, research, and training activities, which is part of the university. So I like to break it up with some pictures, so you're not just looking at a lot of words. So this is just a couple of pictures from our infectious diseases laboratory located in the Baxter 2 building, conveniently located across from Panera, which is kind of dangerous for some of us. But as an infectious disease laboratory, safety of our staff working with clinical specimens and isolates is of utmost importance. So we have our biosafety cabinet certified annually. We have 2 large ones, which was definitely nice during COVID because we could have two people working to process things safely. And then we have some of our assays are immunological in nature. So they're enzyme immunoassays to look for Lyme disease in patients as part of our tick-borne diseases test panel. And we also do a test to look for TB exposure called the quantiferon TB Gold. Some of you may have to do that, on an annual basis to make sure that you haven't been exposed during patient care. So a lot of what we do is molecular testing. We're thankful that PCR has evolved to the point it is. When I started in the laboratory, we were using the instrument on the left that Dr. Summerskill had chosen called the Aries. It was made by a company called Luminex. And it's a sample to answer system, which means we can take a clinical specimen, do very minimal processing, put it on with the primers and probes or however the assay happens to be designed. And then in about 2 hours, we'll have a result of whether we detected a particular target or not. So unfortunately, in late 2023, we found out that Luminex had been bought by a bigger company and had decided they're going to discontinue this platform. And so we had to start the process of, talking to different vendors, manufacturers to see what would be a suitable replacement for this instrument, which had served us so well. We had 5 of them during COVID. We bought a couple of extra to do the capacity of testing we needed to do. So after going through the, procurement process and all that, we ended up with the instrument on the right, which is the BD Max. And so we've spent most of 2024 trying to transition our assays we've been doing for many years with great success on this instrument to a brand new instrument. And it was challenging. I'll just leave it at that. So how do we help with research activities? Well, we provide laboratory testing to support diagnosis in a couple of very specific areas. So we are a niche laboratory. We have tailored our tick-borne disease panel to our region. So we're only looking for pathogens we think, would be commonly acquired here. Our division has had a long interest in community-acquired pneumonia, so respiratory infections. We do a test for atypical causes of pneumonia. We do syphilis screening for a very important program we have in our division, which is the civil surgeon application process, which requires a syphilis test. So we just do that for that specific program. And then we do TB exposure testing using the quantiferon interferon gamma release assay. Not only for that civil surgeon program, but also for hospital systems, because again, employees need to be screened usually on an annual basis. Because our staff are excellent and where they live in the city, they are kind enough to stop by certain hospital locations and pick up samples on their way in. The rest come by courier. If there are questions about our test results or something seems to not match up with maybe what the provider was thinking, we do phone consultations, we talk about it, help them understand the limitations of some of these methods and what they may or may not mean. So really the research aspect comes in when we're trying to develop or implement new testing methodologies that support better infectious disease diagnostics or epidemiological research. So for an example, Streptococcus pneumoniae, we took over this testing when someone retired that also worked at UofL and Norton to provide serotyping of streptococcus isolates that were, obtained from patients with community-acquired pneumonia. And there's an old-fashioned but still wonderful method called the Quellon reaction that allows us to look through a microscope and see what serotype that particular isolate might be. We've done over 200 isolates since we brought in this assay, and we're preparing a manuscript to show all the serotypes that we've been able to see. And so that's going to help vaccine manufacturers that make these, multivalent pneumococcal vaccines to say, this is what we're seeing in Louisville, and perhaps that will help inform them as to maybe what they need to tweak in their next version of the vaccine. We also try to submit proposals to perform applied research in the field of infectious diseases. Before the pandemic, we were fortunate to receive some KBIRN funds for a urine microbiome pilot project, and that's thanks to all the urines we had collected for many years from a study that I'll talk about in a minute. And so we were able to get funding to do that and get some sequence information. So since we're a diagnostic laboratory, I just wanted to give you a sense of the volume of testing we do for some of these assays. So our tick-borne panel, as I said, it includes Lyme disease serology, it includes rickettsia serology, and we do one PCR test, but we get 2 answers for ehrlichiosis and anaplasmosis. Ehrlichiosis is really what we expect to find in this area, but if people travel, there's a chance they may acquire anaplasmosis. These are both tick-borne illnesses. So I'm just showing Ehrlichia because I think we've had 1 anaplasma the whole time I've been here at the university. So we usually test between, 1,000. Last year was a big year. We had almost 2,000 patients that we tested for Ehrlichia. We usually have about a 1 to 2% positivity rate using our PCR assay on whole blood. So once we get the sample, we can have this answer in, 2 to 4 hours. So this is TB exposure testing. This is the quantiferon TB gold assay. It's a gamma interferon release assay looking for, again, it doesn't tell you whether the person actually has TB. It just says they've been exposed and need additional follow-up. So we used to do for us, it's a high volume, over 4,000 tests a year. But one of our big clients decided they were going to bring the test in-house. So that's why the volume has kind of plateaued since they made that decision. But we still do about 2,000 of those a year. And then finally, I just want to show this because it's been a very interesting mycoplasma year. So this is 2 organisms that are on our atypical pneumonia panel. So we accept BAL samples, so lower respiratory specimens, or oropharyngeal swabs, but really lower respiratory tract is better if the person has pneumonia. So we could detect these targets in 1 respiratory sample. And so 2024, you can see, so mycoplasma pneumonia is the blue bar. You can see, and this is just year to date, actually, as of like last week when I was doing these slides, it has been a bumper crop year for mycoplasma. So a lot of walking pneumonia, and it is a cyclical disease from what I understand. So this is just one of those peak years for mycoplasma. And Legionella just is kind of there at a steady state. Physicians can order Legionella only, so we could either do all the targets or we could do single targets, just depending on what the physician needs. So we're pretty flexible in that regard. So since 2016, so since my tenure at the laboratory, we have developed or implemented a number of these molecular sample to answer assays. When COVID pandemic started, we had an assay up and going in March of 2020. And so we were able to do some testing for our healthcare facilities because obviously at that point in the pandemic in March of 2020, healthcare workers were at high risk. So we were offering the test to them. So we were doing a lot of COVID testing in 2020 and 2021. We also, at the request of our physicians, which is really important, and I want to shout out our ID faculty because they're kind of our eyes and ears at the hospitals to let us know that, hey, this test, hasn't performed well, or it's taking too long, which is the most popular reason. It's taking too long for us to get results back, and it's not really helpful in patient management. Could you look at bringing this in-house so we can get a faster turnaround time? So pneumocystis jirovecii is a uncommon pneumonia. It's an opportunistic pathogen. And so we worked really hard to bring that test in-house. It's not common, but when it's on the differential, it's important to have a quick answer. Due to low volume, we don't offer that anymore, but we did have it up and going for a while and it worked really well. Many times these improvements in diagnostic testing or collaborations with other institutions has allowed us to publish these methods so that other people can adopt them if they so choose. And our laboratory has participated in a number of collaborations, and I thought that was maybe the best way to illustrate our role in clinical research or applied research, is just to kind of highlight some of those that led to publications. But the message really is we're open to collaborations. We're pretty much up for anything that our staff and our equipment allows us to do. All right, so one of the first studies we did when I arrived at the laboratory was to support a company in Denmark called SSI Diagnostica. They were developing a device that combined antigen detection of Legionella pneumophila and Streptococcus pneumoniae, so two common causes of pneumonia, into one. There were single tests available. They wanted to combine it into one. And so we did a lot of blinded samples, and we did this with the University of Pennsylvania as another site that also did these blinded tests. And so that resulted in a publication, and it was basically helping this Danish company get US data that they could submit to the FDA. Going back to the PJP assay we did, the target that we had chosen to implement for detection of this pathogen in BAL samples was a large mitochondrial subunit. Colleagues at Johns Hopkins were using a small subunit, so we did sample exchanges and compared our results to see which target worked better. As a result of that, we had a publication, but it was a really nice collaboration, and they were great to work with. So again, COVID-19 took up a lot of our life in 2020 and 2021. There were a lot of research studies going on. Everybody was doing some kind of COVID research at that time, because I think that was the only thing allowed while the campus was shut down. And so I just wanted to highlight one, just because it highlights the collaborative nature of our work. So you can see listed here all the different departments, different centers we worked with to have a publication in scientific reports. So that again was a very positive collaboration with many people at UofL and beyond. Sometimes the studies we do are just internal. So because our division has had an interest in pneumonia for so long, we thought maybe a better diagnostic approach to get a quicker answer is to be able to directly detect this bacteria in clinical specimens. So we worked internally to do this. We had a fellow from Argentina that was spending some months with us, so this was her project. And we ended up publishing it because we felt like it was a really, really good assay to use on clinical specimens. The way we have to approach these new assays, if we want to use it for diagnostic purposes, is we have to meet clear requirements. So we have to go through a lot of experiments to determine performance characteristics. So because of our biorepository, we have samples that were from consented patients to be used for method validations, quality assurance purposes, et cetera. So we can use those specimens in our freezers to spike them with strep pneumonia and do a lot of the analytical studies. So at the end of that, though, we like to know, it's nice to know it works in a laboratory and under controlled conditions and we're spiking everything, but how does it really work on clinical specimens? And that's when we really are thankful for the clinical researchers we have in our division that typically are foreign medical graduates, and they're wanting to get clinical research experience. And so they're credentialed to go to the hospitals, consent patients, collect specimens, or get waste specimens in this case. We needed waste specimens of different types. We did urine, we did BALs, we did blood and CSF because it can present in different ways, not just pneumonia. And so they were really critical into getting these clinical samples so we could really put our assay through the paces to see how well it performed. So to go into left field now, after what I've just told you, during the COVID pandemic, we got to know a researcher in the Department of Bioengineering named Dr. George Pantelos, and a surgeon in the cardiovascular and thoracic surgery department named Victor von Berkel. And they approached us because they wanted to sort of replicate a model that Canada had up with into how to make more lungs available for transplantation. And they wanted to use a pig model, and they basically needed microbiologists to grow up an organism that would give their pigs pneumonia. So we tried Pseudomonas first, and that did not work. So then we went to Staph aureus, and that model ended up working. So this has been an ongoing project. It's very expensive to do, large animal studies. But our job in this project is to keep providing them with Staph aureus when they want to infect a pig. And then as the pig goes through different time points, like day 3, 6, 9, we look to see, they bring us a BAL sample, we plate it on selective media, and we see, is there Staph aureus there and how much is there? So in the top panel, you can see this is a pig that was successfully infected. These are selective media for Staph aureus. So if it's there, it turns the media yellow. And so we look at, day 0, 3, and 6, and 9, and we provide this data. They're also doing some breath analysis, which we're not part of that. But this has been a fun kind of different project to support. So for the ID laboratory, I'm optimistic about the future of the laboratory. We've been in existence for a long time, and whether we're doing more research or more diagnostics, it kind of ebbs and flows. But we're excited to be working on bringing on a test called the Fungitel Stat test, which will help our physicians diagnose invasive fungal diseases. This came to us through, again, one of our faculty members saying, it's taking us at least 7 days to get a result because it's a send out test from the hospital. And, by then by the time we get a result back, we've already had to make decisions about the patient. And so they asked us if we could bring it in-house. So we're in the process of doing that. So when it's live, when the test is live, we'll be able to give them an answer either same day or next day, depending on when we get the sample. Another test that we're looking to bring on in the future is to support our alcoholic liver disease colleagues, Dr. McLean, Dr. Vatsalya, in managing their patients with alcoholic liver disease. This is not an FDA-approved test. And so that means we have to establish all the performance characteristics using samples that they can provide us. These are keratin degradation enzymes, so they're enzyme immunoassays. And this will, the business proposal, we showed that by bringing this test on board, it would help them determine if their patients would benefit from steroids versus being put in ICU and wait for a liver transplant. So we're working on that with them to get that live. So what are the challenges that we have in the diagnostic laboratory? The FDA decided or published in May that they would have the authority to regulate lab developed tests, which is the bread and butter of our laboratory. It's what makes us us. And so this was a sad day for me because I knew that this was going to be a heavy administrative burden and very costly. So there's some lawsuits out there. We obviously have a change in administration. So FDA is in the HHS arena, so we'll see what happens. But basically what we're doing, our approach is we're not going to stop what we're doing. We're going to, do the testing we would do. And then if FDA would require more to approve this test, we'll go back and fill that in. But we're trying to listen to every webinar the FDA does and gather all the information we can. So that's something that's definitely on the radar. And then as always, just predicting technology changes in the diagnostic arena. Point of care testing is greatly desired, has its limitations, but it's getting better and better. They're simplifying these molecular instruments so that they can be done in lower complexity laboratories. And so just trying to keep an eye on where is the field of diagnostics going is a challenge. I wish I had a crystal ball, but I do not. I can stop here. Do you have any questions for me at this point about the diagnostic laboratory? Yes.
I'm running a clinical trial and collecting plasma, serum, and urine from the patient. So how we can work with you to find out some of the circulating marker in urine, plasma, and so on. Is there charge basis facility, work with the investigator?
So, yes, we work it out. Dan Cap is our financial guru and he's here. So, Dan is my friend, so he helps me out. So for the diagnostic tests we do, they're already, live and they're being used at the hospitals. We're a fee for service. We don't deal with insurance at all. So we just have set fees that we bill to the hospital systems on a monthly basis. For studies, we generally we talk about it, like what funding do you have? We worked with Dr. Batsaya to say what's the cost per test to us versus our tech time. And then we come up with some number. And of course, we talk about, can we do it? Is it a test that we can do for you?
Yeah,
So-
some will be PCR based,
Yeah.
some will be ELISA based and so on.
Yeah, so we can definitely have a conversation and we'll connect, but it starts with a conversation, just saying like, what do you need? What do we have? And, does it make sense for us to do it for you?
Okay, great.
All right, any other questions?
Well, this, at the bottom of one of your slides, you had the urinary microbiome.
Yes.
I thought that the urine should be sterile, so that you're only talking about urinary tract infections that you're measuring?
So what we did is, again, because we're interested in pneumonia, and one of the antigen tests you can do is a urine dipstick, or not a urine dipstick, but it's a urine test,
Okay,
urine antigen test. So we have a lot of urine. So we're like, what resources do we have? And we just have to select the samples from our freezers that
okay,
we could use for this urine microbiome. Yeah, so in the world of microbiomes, we're finding that things that we thought were sterile are not actually sterile. So what we did is we compared like healthy people to people with either like influenza or community-acquired pneumonia, and we just tried to see, is there a difference in the genus of bacteria that are present? Is there a difference? And unfortunately, that was right before COVID, so that has been backburnered. Like, we have the data, but we haven't been able to do anything with it yet, so just to be
Well,
honest.
groundbreaking stuff. If there are people are running around with colonization and a normal versus abnormal
Yeah.
microbiome and what changes it and what
Right,
pathologic events
Yeah.
happen after.
yeah, it's really interesting. But yeah, unfortunately, just to be honest, it got backburnered to COVID, yes, for us.
You had some graphs where you showed how many tests you do. One was for the quantiferon, one was
Mhm.
for tick test. Can you estimate off the top of your head how many COVID tests you did over the last few years? Obviously, in 2019, it'd be zero.
Yeah, 2019 it was 0. In 2020, it was, I know it was our highest volume test, so it was probably 10,000, just COVID. So when I looked at our volume, it went from like 0 to like this, because at some point people didn't care anymore. So no one was
Yeah.
at
There was
doing-
a diagnostic laboratory,
Yeah.
was going, Quest was going.
Right,
Yeah.
So
Yeah.
there was a different, at the
Yeah.
beginning, I think.
Right.
This laboratory was the only one.
yeah. And I have to keep it separated because my health department laboratory was also doing it and we had the same thing, it went move down. So, but I know it was because it was pretty much the only thing people were doing at that point.
Yeah, an
in the
impressive
pandemic.
number though.
Yes,
Is worth
yes,
telling.
yeah. So sorry I left that one out, but it's probably just, me repressing all those memories. Alrighty, so now I'm gonna turn and do my best to channel Dr. Subi. So she's our director of the Infectious Diseases Biorepository, which was started before I came. And she's been involved with the Biorepository since before I came. So the goal of the Biorepository is basically to process, store, and release specimens from consented patients and maintain sample integrity at all times. So our biorepository is also unique because every sample that we have stored, well, except for maybe some COVID, because that was crazy, but for everything else, we have clinical data that is associated with every sample, and we know where every sample is. And so this is not only useful to our own internal ID studies, but it could be useful to other people who are looking for urine samples or respiratory samples if they want to look at some other biomarker or something like that. And so we have all that metadata that goes with the patient. So the biorepository by its nature supports clinical and applied research. It also allows our research interns, so these foreign medical graduates, to get experience in the laboratory if they are interested in that. And it just helps build their resume if they want to continue to practice medicine in the United States. So just a snapshot, the biorepository is really about our -80 freezers. We have 9 of them. And they all have monitors on them to make sure that they stay within temperature, within range. If they are not, if something happens, then Subi and I both get a text and an email to let us know that something's not right so we can address it. So these freezers are full of, as I said, a lot of different clinical specimen types. We have a lot of urine because of all the studies we've done over the years. So every tube you see here has a number on it. We know what space it's in the box. We know which rack in the freezer it's in, what shelf of the freezer. It's all in our REDCap database. So if we need certain specimens, we can find them and give them to other researchers or use them ourselves if we need to. So as I said, they help the ID laboratory diagnostic side perform applied research. If we need a certain sample type, we can go to the freezer and spike it with what we need. We have an implementation team that we work with when we have a new study that we write. We help them determine the correct collection procedures, how they're properly transported to the laboratory, our protocols. Once it's in the laboratory, what do we do? And because of our record keeping, we can answer any queries that a sponsor might have about a sample. We have a database management team because all of our studies are in REDCap. And so for each study, they create a specific instance or page for us in REDCap. And everybody gets trained on how to enter data in that. Quality assurance team helps reconcile lab sample data. And then again, because we're using human subjects, it's really important that we have a great relationship with the clinical trials unit so that we're compliant. So we make sure our IOBs allow for consenting of patients, as well as the specific new protocol we're going to follow. So the workflow in the biorepository is, once we receive notification that someone wants to do a study, we make plans to make sure we have everything in place. We write the procedures, we buy any necessary supplies for it. We train our lab staff on how to perform the procedures on the specimens that will be coming into us using good lab practice. We process, label, catalog, and store the specimens. We enter sample data into REDCap. We answer queries. If someone else needs the samples, we release them and update inventory so that we know what's been kept and what's been sent. And so record keeping is a big part of the biorepository. And as I said, because we have these ultra low freezers with all these precious specimens, they are monitored 24/7. So what does it look like? I'll just pick a simple example. Since so many of our studies involve urine from community acquired pneumonia patients, we'll get urine brought to us by a research intern. And this is all like on a time schedule because the sponsors usually have a really set time of when the sample is collected and when it has to be at the laboratory. And we add a buffer to stabilize it. We mix it well, and then we aliquot it into these tubes, label all the tubes, put them in boxes, store them, all that good stuff. So when I first got here, one of the large studies that was done with Pfizer around Streptococcus pneumoniae infections was called urinary antigen development 1, and then 2 followed. So these are done periodically, again, to kind of check their vaccine efficacy. So these 2 studies were done in adults 18 years and older that had confirmed, radiologically confirmed community-acquired pneumonia, so not ventilator-associated pneumonia. So as I showed you in the graphic, we processed all these urine samples that came in. We shipped them to Pfizer because they had another specialized test they wanted to do on these. And then isolates that were available from patients with these infections were also grown and stored and shipped to Baylor College of Medicine at that time so they could do the serotyping. Back then, we did not have the ability to do the serotyping. And then some were actually shipped to Germany, which is always fun to do international infectious disease shipments. UAV2 was similar. And so as a result of this work, we had a nice paper published in Clinical Infectious Disease Journal that was considered a major article. And the reason I wanted to show this is not just to show we get publications out of these studies, But just to make the point that it turns out that Louisville is a really good representation, demographics and other factors for the rest of the United States. So if we could do a population-based epidemiological study here in Louisville, then they can make extrapolations to the rest of the United States. And so we think that's, part of the reason that these vaccine manufacturers like to come to us to do studies, not just because of our experience, but also because of our population. So we have a current study going on, again, just sort of following up on the pneumococcal vaccines. And so they're just trying to see like what's the efficacy of the most recent version of these vaccines. This is only being done in adults 65 years and older, just because that's the recommendation for these vaccines. And again, we're getting urine from these patients. If we can get an isolate, we do get those and store them. We're also sending them to Baylor because at the time this started, we weren't up and going with the Quellon reaction. So talked about urine studies, which is, we have a lot of urine. We did another epidemiological based study around C. difficile, which is a big concern in hospitals. CDC is concerned about it because it tends to be a multi-drug resistant organism. So this is an active surveillance system. And in this study, our lab processed 1,400 plus stool specimens for people that met the criteria for having a certain consistency of stool. And so we did a rapid antigen test to see if the toxin was present in the person's stool. If it was, then those were the samples that we aliquoted and shipped to a Pfizer lab in New York for them to do some specialized testing on to look for cytotoxins. And we kept some of the positive stool samples on site as well. And so if we have these additional samples that Pfizer didn't require, then in this case, it allowed us to do a co-infection study. We were curious in adults, were any of these people that had C. diff also infected with something else? So what we found is mostly they had some E. coli. That's about it. So again, this led to a publication about standard of care specimen collection and how people may be misdiagnosed. At the time we started this study in 2019, again, it got interrupted by COVID. So we had to stop and start it. But the recommendations for doing C. difficile testing in hospitals, some were doing it one way and some were doing it another, pros and cons to the algorithm. But basically the standard of care specimen collection, we estimated that they were misdiagnosing some patients with C. diff. So as I said, we did a lot of COVID-19 related studies. So in this case, colleagues in the Department of Chemical Engineering and Anesthesiology and Perioperative Medicine received NIH funding to see if there was a non-invasive way to diagnose patients with COVID. So they were doing a breath analysis study. And you may think, why in the world were you involved in this project if you're looking at breath? Because we're an infectious disease laboratory, we deal with specimens and isolates. Basically because it was COVID and they were engineers, they did not want to touch anything that might be infectious. So they collected the breath and brought us the breath bags, and we eluted the air out of the bag onto a chip, which they then took back to their laboratory and did the fancy HPLC and all that good stuff. So that was our role, was to process the breath bags and elute them. So it was definitely a very different project for us, but we were happy to be able to support them in their research. So that also resulted in a publication with them. It was fun and interesting because it was different, but we were approached by a vaccine manufacturer, Pfizer, who really wanted to understand what is the carriage rate of Neisseria meningitidis in college students, because they're recommended to get a vaccine because of their age and living in a dorm and being in close proximity, and how dangerous this infection can be, how quickly they can decline if they're infected. So basically this was like, what's the carriage rate? So we had to work with campus health and a lot of people on Belknap campus because they really wanted to capture swabs from these kids who were just moving into the dorm. And they wanted to sample them at 3 time points to see did their carriage rate change over time as they were in close proximity to other students. So there were 3 sampling time points. Neisseria meningitidis, there is not a media that you can buy for this. So we had to make bottles and bottles of this media ourselves. So we got a recipe, we made it, and it worked very well. And so all of those were those samples that were collected by our interns at different sites around Belknap campus were brought to the laboratory. It's a very fragile organism, so we had to do all this really quickly. So it was kind of like a factory for a while in our laboratory while we were doing this study. But they were frozen as quickly as possible and then shipped to a central laboratory for them to actually do the work on the organism and do some characterization. And so that manuscript is in preparation. Thanks to COVID, we got involved in some vaccine trials. So from 2020 to 2023, we worked with Johnson & Johnson on their COVID vaccine study. The role of the laboratory really there was to, make sure they didn't have COVID to start with, but if they did develop signs and symptoms of a respiratory disease, that we would do some testing to see what they were sick with. And very similar for RSV, there's a push for RSV vaccines for adults, especially if they have underlying conditions like COPD or something. And so we were involved in a phase 3 efficacy study for a Johnson & Johnson RSV study, which ended last year. Another collaboration we've done on the biorepository side was looking for SARS-CoV-2 colonization and looking at characterizing the antibodies from these people that were infected. And so we had to enroll people that were asymptomatic as well as symptomatic to, make some interpretations about the data. So that was another UofL internal collaboration. We've had a post-COVID research clinic going since the pandemic. Dr. Huang, again, in the Department of Anesthesiology and Perioperative Medicine, got a grant from Gilead Sciences to really look at cardiovascular and pulmonary long-term effects of people who had, long COVID. So in this study, we're getting whole blood, plasma, serum, urine samples to be stored for future research. So that's ongoing. And then finally, the study we're doing now, which is Dr. Kavalazzi here at UofL plus Duke, we're looking at ventilator-associated pneumonia. And so when we get these samples, again, we have to be available because if there's a clinical change, then we need another set of samples right away. This is all, on a time clock. So our staff are a little bit on call when we hear about a patient who's been enrolled in this. But in this study, we're getting endotracheal suction since they're on a ventilator, leftover BAL samples, and any clinical isolates that came from respiratory samples or blood from the microbiology laboratory. So we basically collect and store anything and everything. So finally, I'd just like to acknowledge the team we have. We have a great team in our division. Here's a picture of us. We actually occasionally socialize outside of work. So we went to the melting pot and had a nice dinner. So this is our 8-person lab team. This is Dr. Subi, who couldn't be here today. Dr. Arnold, our division chief, he's just a big supporter of the laboratory, which we so appreciate. Dan Kapp, he keeps after our finances and keeps us on the straight and narrow. Steve Goody is in charge of operations and research. So for all these sponsored projects and other projects, he's key to that. And then we have a couple of research managers that coordinate the research interns and train them and make sure they're doing what they're supposed to do for all these studies. So Vigio and Danya are really important. And then Marita has historically been our designee from the clinical trials unit to help us with all the many IRBs that we have going for all of these studies. I think that's my last slide. So thank you all. I'm happy to answer any questions.
Biorepository researchers, it's really good to know they have a capability.
Yeah, it's definitely an infrastructure we've built over time, and so now it's kind of like a well-oiled machine for the most part.
Well, thank you very much. It's good to know what our capabilities are, and I'm sure that'll stimulate some collaborations within this room.
Yeah.
Thank you.